Now I'd like to introduce today's speakers, Dr. Nora Besansky and Guiyun Yan. Nora Besansky is the Martin J. Gillen Professor of Biological Sciences at the University of Notre Dame. She's also a member of the National Academies of Sciences. She's an expert on the evolutionary, ecological, and functional genomics of malaria vectors. Nora played leading roles in coordinating two large international genome sequencing and analysis projects that were sponsored by the National Institutes of Health that were directly responsible for making valuable genomic resources regarding malaria vectors available to the research community for more than 16 malaria vector species. She continues to be a leader in vector genomics. Her research is driven by a desire to understand why and how the transition from non-vector to vector has occurred, which has happened repeatedly during the evolutionary diversification of Anopheles mosquitoes. Guiyun Yan is Professor of Public Health at the University of California, Irvine. Guiyun's research interests are the biology of mosquito vectors of human disease and epidemiology of vector-borne diseases. Guiyun's research interests are the biology of mosquito vectors of human disease and the epidemiology of vector-borne diseases. Guiyun has a very international research program that has included studying the ecological mechanisms of malaria epidemics in high elevation areas in eastern Africa, the impact of environmental modification on malaria epidemiology and transmission, and new vector control strategies. He also works in Asia, where his work involves investigating the mechanisms of heterogeneous malaria transmission in the border area of the greater Mekong region, and the development of integrated malaria control strategies to aid the malaria elimination efforts in that region. Guiyun has recently been conducting population genetic studies at temporal and spatial scales that are much finer than those typically associated with population genetic studies. This is referred to as landscape genetics, and we'll hear more about that today. I'm very excited that both of you are able to join us today, given your busy schedules. Nora, you'll be our first speaker, and I'm ready to turn the floor over to you.
Thank you very much for that kind introduction, and to Hector and also Stephanie James and the other organizers of this event. It's a pleasure to participate in it. Thank you very much. So my topic today is the likelihood of gene flow between Anopheline malaria mosquitoes. And I have divided the talk into these 5 parts. So let's just go ahead and get started with the first one, where I'm providing some basic background to the problem. So apologies, because this is being pitched to a non-specialist audience. So, experts, just, I hope I don't bore you too much. Okay, so the first thing to note is that interspecific gene flow can occur in 2 fundamentally different ways. So the first way is horizontal, sometimes referred to as lateral transfer of genetic material. And this refers to non-homologous exchange between non-mating species. So this is the dominant mode among prokaryotic species, but it's incredibly rare in eukaryotes, even on an evolutionary timescale. So examples that you might be familiar with would be the acquisition of mitochondria and chloroplasts from bacteria during the emergence of eukaryotes. And another example you might be familiar with is the horizontal introduction of transposable elements and viral sequences into eukaryotic germlines. I mean, today, with all of the genomic sequencing that's going on, we know that transposable elements and viral sequences comprise, in some cases, very large fractions of the genome. So in the human genome, transposable elements occupy about 45% of the content, which is kind of crazy. But despite all of that, the actual rate of horizontal flow, transfer or gene flow is very low. So we're talking about one event per transposable element family per 20 million years based on estimates from Drosophila. So this is what I mean when I say incredibly rare in eukaryotes. And this is in distinction to vertical transfer, which is also known as introgressive hybridization. And that's the method of interspecific gene flow that's probably more familiar to most of us. That involves interspecific mating, and that's the mode that's most common in eukaryotes, and therefore will be the main focus of this talk. Another basic point to drive home is that hybridization is not the same thing as introgression. So hybridization is just interspecific mating, but it doesn't inevitably lead to introgression. And a familiar example is shown here. We know that horses and donkeys can hybridize, and the offspring is fertile. It makes a mule, but mules are sterile, and therefore this hybridization does not have any genetic or evolutionary consequences. Another example that may be more close to home for at least some of us is the hybridization that occurs naturally between a couple of culisine mosquitoes, namely Aedes aegypti and Aedes albopictus. These are not very closely related species, actually, but they do hybridize in nature. In this case, though, viable offspring are not even formed as a result. So for sure, there's no genetic or evolutionary consequence of this event. It's not to say there aren't ecological consequences, but there's not any genetic or evolutionary consequences. So when we talk about interspecific gene flow, we're really talking about introgressive hybridization, known as introgression for short. And that's defined as the transfer of genes between species through backcrossing of viable and fertile F1 hybrids. So backcrossing to one or both parents. So this phenomenon was defined by botanists decades ago, in the 30s. Well documented and very well accepted by botanists. I forgot to say, I want to flip back. I know why I forgot to say it, because my gallery view was hiding it. So I want to say that throughout this talk, I'm going to have superscripts that refer to literature that I've cited and will cite throughout. And there's a slide at the end. There's a slide at the end of the PowerPoint deck that lists those citations for your reference. You'll be able to access it in the recording. So, okay, my mouse is jumping around, forgive me. I'll try to control that. All right, so the point I was making here, and this refers to reference number 2, excuse me, is that introgression was defined by botanists a long time ago. It's been documented by them extensively and was very well accepted historically. And this is in distinction to the acceptance of introgression by zoologists. And the basic reason why acceptance of introgression among zoologists was so slow was because of the dominance of several individuals, including two shown here, Ernst Mayr and Theodosius Dobzhansky, who were in the 20th century promoting this biological species concept, which sort of it dominated the a view of species that was as follows. Species were groups of individuals that can interbreed but are reproductively isolated from other such groups. So under this biological species concept definition, hybrids were viewed as unnatural, first of all, very rare, and if they occurred, they would be evolutionary dead ends due to being unfit, or the expectation that they would be unfit. Now, we know today that this was an extreme view that really is not correct. We now know that introgression is widespread across the tree of life, not only in plants, but in animals. But in fairness to those who didn't believe it in the last century, we have to remember that, the evidence for historical introgression, which I'll define in a minute here, is not usually apparent to the naked eye, and even from a genetic standpoint is basically impossible to detect if you have only one or a handful of markers. And let's not forget that the human genome was not sequenced until 2001, using what we now view as very antiquated technology. And so, the tools available to zoologists in the last century didn't allow the revelation of introgression that we now see based on widespread whole genome sequencing using second and third generation technologies, accompanied by the huge increase in computational power and algorithms for analyzing these data. So with all of that, the studies that are revealing historical introgression, which is evidence of past gene exchange, whether or not it's still happening in contemporary days, those numbers of studies are growing literally daily. So it's a partial listing here with appropriate references that you'll find at the end. But I'm sure all of us have heard about the introgression that was reported between anatomically modern humans and Neanderthals, as well as Denisovans. But all of these other examples and many more have been documented in the literature. Well, so some of you may be thinking at this point, this is not what I learned in high school. Animal species are supposed to be incompatible with one another, aren't they? Well, no, not immediately, right? So when we think about animal species being incompatible, what we're really talking about is intrinsic genetic incompatibility in the form of either hybrid sterility or inviability, and that takes a long time to evolve. So the hybridization events that we're talking about here are typically among very closely related species. And it turns out that there's a window for that kind of hybridization and compatibility that goes out up to about 5 million years in insects and longer than that in other animals like birds. So what enforces isolation before that intrinsic genetic incompatibility evolves is actually things like behavior and ecology. And those vary in space and time. So, very locally, ecology changes, behavior changes, and depending upon the environmental conditions, hybrids that form may actually be just as fit as the parents. So again, incompatibility takes a while to develop. So early on, the chances are that a large fraction of the genome between different species closely related is in fact compatible. So introgression, it turns out, is especially prevalent in species radiations. So what is a species radiation? It's simply a rapid diversification from a single ancestral species. So shown here are figures that I took from the literature, and these are sort of charismatic examples of species radiations that I'm sure all of you are familiar with. So the cichlids of the African Rift Valley lakes, Helicones butterflies, Darwin's finches. So by definition, species and radiations are very closely related, and soon after their emergence, you'd expect them to be in close geographic proximity. So the opportunity for introgression is there, and it may simply occur because of opportunistic phenomena. But what is coming out more and more in the literature is that introgression may be more than simply opportunistic. It may actually be responsible for driving further radiation. I'll touch on that in a bit. But importantly, nearly all malaria vectors that we know are part of species radiations. So that brings me to the next part of this talk, which now gets us into anophelines and historical introgression among them, using as examples the Anopheles gambiae complex and the Anopheles funustis complex, which happen to contain the world's most important vectors of malaria. So let's get started first with the Anopheles gambiae complex. So this is a group of at least 8 morphologically identical species that radiated, according to the most recent estimates, less than 500,000 years ago. So among these 8 species are 3 that are considered major vectors. Arabiensis is one of them, shown in the red in panel A. So you can see it's very widely distributed. Anopheles gambiae and Anopheles colutzii are shown jointly in green in panel B. Those are also very, very widely distributed. These vector species are highly anthropophilic, especially gambiae and colutzii, and endophilic, indoor resting, indoor biting, and not coincidentally are very polymorphic, both in terms of their genome nucleotide diversity and in terms of chromosomal inversions. The other 5 species are minor vectors or even non-vectors, and you can see in panels C and D that they have much more restricted ranges. They tend to be zoophilic and exophagic and are characterized by much lower levels of polymorphism. Now, their interbreeding between any of these species pairs is possible, and reproductive isolation is incomplete in the sense that the hybrid females, the F1 hybrid females, are viable and fertile and can backcross in either direction. So introgression is at least plausible. Now, as Dave mentioned in the introduction, I and many others were participated in a major genome sequencing effort, the fruits of which were published in 2015. So we performed whole genome sequencing on at least 16 different anopheline species and assembled reference genomes and other genomic resources. And in one of the publications in 2015, we focused on the gambiae complex and its closest relatives to try to infer the historical branching relationships among the species, a question that had been long unresolved, and to try to understand whether introgression had played a role in the evolution of these species. So our analysis was based on genome alignments between, I'm not going to bore you with the species names, but 6 members of the gambiae complex and the closest relatives outside of that complex. And out of this analysis came the revelation that there had been two major introgression events shown here in the green arrows between different species pairs. And importantly, one of the most massive of the introgression events was from arabiensis into the lineage that gave rise eventually to the species gambiae and collutii. And we argued that that the origin really of gambiae and collutii as major vectors was probably not accidental, but was as a result of introgression from arabiensis, because alleles were introduced that conferred aridity tolerance, which probably led to their range expansion that you saw in green on a previous slide, that sort of led to a huge range expansion, and probably also the capacity to track humans in anthropogenic environments. Importantly, one of the tests that we use to infer introgression, which is known as ABA-BABA, detected no introgression outside of the gambiae complex. So despite all of these introgression events within the radiation, we did not detect any sign of introgression between any of the gambiae complex species and the closest relatives outside. Okay, so now let's talk a little bit about the funustis complex. And you'll notice that there are very striking parallels here, even though these are very different species. So this complex comprises 7, or I should say at least 7, morphologically similar species. Again, a very recent radiation. And again, this theme that in this case, there's only one major vector. The rest are very minor or non-vector species. So the major vector, funustis itself, is widely distributed, and that's that big swath of red that you're looking at. The vector species is highly anthropophilic and endophilic and polymorphic, whereas the vectors, minor vectors or non-vectors, have much more restricted ranges. They're zoophilic, they're exophilic, and they have much reduced polymorphism. But just like in the gambiae complex, cross hybridization is feasible among all the species, and introgression is plausible because the F1 hybrid female is fertile, at least in some crosses. Okay, so much more recently, we took a look at introgression in the funustis complex. And to do this, we had to do some sequencing and assemble some new reference genomes for members of the funustis group other than funustis itself. This is an unfortunately understudied group relative to the gambiae complex. We applied some newer modeling approaches to jointly infer phylogeny and introgression in this particular group of species. And 3 introgression events that we inferred are shown here in green. There's actually a 4th one that we inferred. So there have been, in the very recent history of the radiation of this complex, there have been 4 massive introgression events between different species pairs. And the most notable of these involves distant lineages. Okay, so that blue arrow is pointing to a species that we call parensis. So we inferred an introgression event from, mainly the direction of this was from parensis into the lineage that gave rise to funustis about 13,000 years ago, and we inferred that this predated the expansion of funustis across its current range in sub-Saharan Africa. So here again, we see that introgression is playing an adaptive role in facilitating the range expansion and likely also the, what do I want to say, anthropophilic tendencies of funustis. So it's range expansion and, if you'll forgive this sort of loose terminology, probably also vectorial capacity. Here too, it's important to note that we performed the Abba-Baba test. This was not part of the 2020 publication. This was much more recently and so unpublished, but we applied the Abba-Baba test to see if we could find any evidence of introgression between funustis and its closest relatives outside of the complex, Demyelinae rivulorum minimus, and we found no such evidence. Okay, so this is the fourth part of the talk, and I note here that we've been talking up until now about historical introgression, not necessarily contemporary ongoing introgression. But yet, in the context of regulatory issues surrounding gene drive, it's really contemporary introgression that we're most interested in. So what can we say about that? So first, here's some information about the gambiae complex. So many groups have reported finding F1 hybrids in their field surveys of Anopheles gambiae. And rather than my trying to summarize those, fortunately, there has been a relatively recent publication that comprises a meta-analysis of almost 100 field studies over about 50 years and 25 African countries. So it's a really nice summary. And from this, I've derived the data on this slide. So the prevalence of hybrids between arabiensis and either gambiae or collucii is about 1 in 5,000 across the continent, both in terms of sampled adults and sampled larvae. The prevalence of hybrids between the sister species gambiae and collucii is higher, as you would expect, because they're much more closely related. And that is about 1 in 500 to 600 when we exclude a known region in far West Africa where hybridization rates are exceptionally high. It's important to keep in mind when we think about contemporary hybridization that this is a phenomenon that varies not only sort of country to country, but also really at a local level due to ecological variations, seasonal changes, and so on. And the hybridization rates are positively correlated with the degree of contact between parent species. Also, that stands to reason, right? And so it's highest when the species are at equal frequencies. Now, with the funustis complex, with the caveat being that this really is understudied relative to gambiae, to the best of my knowledge, hybrids have never been reported among species in the complex, at least based on the typical rDNA diagnostics that are performed on adult females as they're collected. There is a single exceptional publication which reports frequencies that, frankly, are just too high to be credible. Frequencies as high as 12%, and of which 6% are hybrids between, purported hybrids between funustis, for example, or another species in the complex, and outgroups outside of the complex. The inference of these hybrids was based on so-called bands on a gel that were not sequenced. So the evidence is not terribly strong, and that inference flies in the face of the Abba-Baba test that I just mentioned to you. So I think there's reason to question this report. So I conclude that evidence for contemporary hybridization within the funustis complex is really lacking right now, but of course it can't be firmly excluded. Negative data is always hard to firmly exclude. Okay, so what about practical implications for gene drive? All right, well, as you've heard, spread within the gambiae species complex is possible. So, the rates of hybridization and introgression are low, right? 0.02% or 0.17%. So they're low, but they're non-negligible. And when you think about it, the most probable events are going to involve the species that are most abundant and the most widespread, which just so happen to be the vectors themselves. So I would argue that this sort of spread would be to the benefit of our trying to control malaria transmission by the vectors in this group rather than viewed as a risk. Now, with respect to the funustis complex, it appears based on available data that spread outside of funustis within its closest relatives is doubtful, but again, there's just a paucity of data, I would say. So there's some question as to whether we should be looking at this in more detail, maybe even just trying to ask the question of whether indeed we have fertile F1 hybrids, even just in the laboratory, which hasn't been looked at as carefully as it has in the gambiae complex because funustis and relatives don't have a history of colonization in the laboratory. They're very difficult to colonize. Now, what about outside of these complexes? So the closest known relative to the gambiae complex is a species called Anopheles christii. As best as we can judge, and those of you who know how we do this realize that there are huge error estimates around our attempts to date species divergences, but we think that christii diverged from the ancestor of the gambiae complex much more than 5 million years ago. Which is a long time in terms of ability to hybridize. And there's no evidence of introgression based on the genomic data, as I mentioned to you. Now, as far as the funustis complex, the closest known relative is demyelinae. I didn't mention it in any detail in the previous slide, but we actually did sequence this species and constructed a reference assembly and conducted Abba-Baba tests, which gave us no indication that there had been any historical introgression between funustis and species like demyelinae, rivulorum minimus outside of the complex. So, and if I now refer to the risk of horizontal spread, because up till now we've been talking about vertical, that's even less likely, to the point where I would say that it's effectively nil, simply because there's the rarity is 1 event per 20 million years. So we cannot rule out the possibility of gene flow within interspecific gene flow within these complexes, but the risk for transgenes or any sort of gene flow to occur between members of the complex and any known species outside seems remote. Okay, so I point out here are the references for anybody who wants to go look it up in more depth later. I point out too, for those of you who want further background than what I had time to cover today, here are a couple of references. And finally, I want to say thanks to a huge team of people who really did all this work. I just sort of waved my hands over them and blessed them. So also thanks to the funders. So that concludes my presentation. And I'd like to turn the floor over to Guiyun.
Great, thank you, Dave. So Wanda, thank you, Dave, Hector, and Stephanie for organizing the Gene Convene webinar series and for inviting me to share our recent study on assessing the impact of landscape features on the gene flow of malaria mosquitoes in Kenya. So I also thank you for coming to this webinar. So here's my outline. So I will briefly talk about gene drive, then the need to assess the impact of landscape features on gene flow. Then I will talk about the application of landscape genetics approach to the study of malaria vectors in Kenya. Finally, the conclusion in the future work. So some of you have seen these slides probably many times. So in the past 2 decades, the scale up of malaria control measures have produced great success in malaria control. So global malaria incidence rates was decreased by 30%. Malaria mortality fall by 6% over this period. Despite these progresses, malaria burden remains high. So for example, in 2019, global malaria clinical case number was estimated 222 million with 384,000 deaths. Among them, 94% deaths occurred in Africa. So particularly worrisome is the progress in malaria control has stalled in the past 5 years in Africa. So as shown here with little change in the mortality rates and in the malaria incidence rates. So the recent COVID-19 pandemic adds additional challenges to malaria control. The main challenge with malaria control and the elimination program is that the residents are being frequently reinfected. So there are multiple reasons for this, including the audit, vector ecology, behavioral insect resistance. The most anti-malaria drugs are not effective at killing gametocytes. Recently, the RTSS vaccine is being tested, rolled out in 3 countries in Africa. So this vaccine has modest effect on clinical malaria and severe malaria, but it is unlikely to have a major impact on transmission because this vaccine is intended for young children, and the young children constitute only a small proportion of the infectious reservoir. So key to control malaria infection is to reduce transmission in a cost-effective and sustainable manner. 2 fundamentally new tools are regarded as game changer for interrupting malaria control. The first is transmission block vaccine that inhibit malaria process development within the mosquitoes through targeting mosquito middle guard or gametocytes. The second is gene drive technology that spread the anti-process genes or mosquito killing genes much faster than the normal inheritance. For this presentation, I will discuss issues related to the spread of gene drive in the field. Ethan Beer from UCSD and Tony James from UCLA conducted pioneer research in developing mosquitoes equipped with gene drive. In 2015, they developed Cas9 mediated gene drive system in Anopheles stemicide and further demonstrate that the pest resistant gene can be spread readily in the target population in the laboratory very rapidly. So normally, organisms have a 50% chance passing along a gene to the offspring. So under the normal inheritance, the other genes will not spread. However, under this gene drive inheritance, the gene drive copies and pastes itself onto chromosome from both parents, so insurance get passed on with more than 50% of chance. So the outer gene can be spread in the population rapidly. Gene drive mosquitoes are being explored for the control of vector-borne diseases. So a vector control research consortium called Targeted Malaria, so as shown in Nora's slides earlier, So Tachytic malaria conducted first release of non-gene drive genetically modified anopheles gambiae sterile male in Burkina Faso in 2019. So to examine the dispersal and the mating behavior of genetically altered mosquitoes. They plan to test gene drive mosquitoes in Burkina Faso, Mali, and Uganda. So lately, so 2 weeks ago, Oxitec, in collaboration with the Florida Case Mosquito Control District, started the first release of genetically modified female killing Aedes aegypti mosquito in the US. Their genetically modified mosquito has no gene drive mechanism. So one important challenge for the gene drive mosquito program is finding a pre-release strategy to rapidly spread the altered gene in natural populations. A research group from Oxford University modeled the spread of so-called driving Y chromosome in Burkina Faso and the surrounding countries. Here, driving Y chromosome is a gene drive technique for population suppression through male-biased sex spatial. The figure on the left shows the scenario of spatially uniform release. The figure on the right shows the time taken for the gene driving Y chromosome to arrive. So their results shows that the consequence of driving Y chromosome introduction vary greatly across the landscape. So most rapid spread occurs in densely populated parts of the landscape, where dispersal between neighboring neighboring settlement is frequent. Given that landscape plays an important role in mosquito dispersal and gene flow, so we need to assess the impact of landscape features on the gene flow of malaria mosquitoes. Mosquito short dispersal is typically within several hundred meters. So these disperse for suitable resting sites, finding mates, blood meal, and the breeding habitats. Using arrow sampling, a study by T.B. Lam's group at the NIH provided evidence for windborne long-distance migration of Malaria in West Africa. So their results suggest windborne dispersal of Anopheles mosquito for several hundred kilometers. So in addition, human-assisted dispersal occurs along the routes with busy traffic. So short distance and the long distance dispersal is impacted by ecological process related to various landscape features. We can use population genetics to detect genetic signature from past dispersal. For example, here we have two populations, population A and B, that have been isolated from each other for a long time by some barriers. We can tell this because these two populations are genetically different from each other. So let's see now the geographic barriers separating these two populations are removed. Mosquitoes are now able to migrate, travel freely between populations, mating with each other after migrating. So after some time, the two populations start to look genetically similar. So we think that these populations are well connected. Geographic barrier may lead to genetic differentiation between populations of close physical proximity. In this hypothetical example of reindeer, let's say the deer's body color is determined by two alleles. So in this part, the deer population is in random mating, no spatial genetic structure. Now, after a highway is constructed, if the highway completely blocks the dispersal of deer population, we may see two genetically distinct populations if different body colors are favored on the different sides of the highway. So therefore, we can have a genetic differentiation among populations that are geographically near to each other. Traditional approach has been used to detect the effect of geographic distance on population genetic structure. And these studies, the isolation by distance approach has been used for malaria in Africa. And these studies found that populations of Anopheles gambiae in Western Kenya were genetically more similar to the populations in Western Africa. So the authors posted that the coast of Kenya were actually more genetically distinct from Western Kenya is because the Rift Valley, this which bisect the country. So the weather inside Rift Valley is hot and dry, no enough gambiae. So the western side of the Rift Valley is surrounded by high plateaus and the mountains, which may limit the dispersal of malaria vectors. So using a traditional isolation by distance approach, we cannot press out which factors are driving this genetic structure pattern. However, using a landscape genetic approach, we can test the association between ecological variables and the genetic relatedness to determine the importance of key ecological variables influence malaria vector dispersal and the gene flow. This approach involves an infer population movement from the distribution of genetic variants. So here, for example, we have three populations, population A, B, which are genetically similar, and population C is genetically distinct from the other two. Now we can put them onto a hypothetical grid landscape. So then we can quantify ecological factors hypothesized to driving dispersal. So in this example, we have forest. So here we might guess that forest serves as a barrier to gene flow. So with more populations, we can statistically test the relationships between gene flow and the ecological factors. The landscape genomics approach involves three approaches, three steps. So first is to measure genetic variation. So this is the same example I used. So we have three populations. Population C is different from the other population too, but A and B are quite similar. So then the second step is to quantify landscape heterogeneity. Right? So here we can overlay the landscape variation above the genetic variation. So here I use the land cover. Right? So the third step is to statistically test the association, the link between genetic variation and the landscape heterogeneity. So there are several statistical methods for this linked-based analysis. So I list them over here. So such as partial mental test, multiple matrix regression risk boundaries, linear mixed effect model, and the Bayesian approach. So this landscape genetic analysis will enable us to quantify the effect of landscape on gene flow. Landscape genetics has mostly been used in the field of conservation biology. So to illustrate usefulness of landscape genetics, so I'd like to provide a couple examples. So here, the approach was used to identify factors associated with landscape resistance to dispersal near endangered monkey species in Tanzania. So this study found that crop fires for making waste for crops and pastures inhibited the monkey gene flow. So on the figure on the right shows a positive association correlation between the frequency of fires and the resistance to gene flow. So higher resistance value lead less gene flow. So furthermore, here, landscape genetics can also be used to identify important migration routes for genetic connectivity. So in this example, researchers have identified putative movement corridor of wild tiger in India. So as shown by this light yellow path. So yeah, this path. We borrowed this approach from conservation biology to improve understanding of malaria vectors. So we're interested in landscape factors either promoting or limiting the gene flow of malaria vectors, so to inform new vector control measure implementation. Here we focus on Anopheles gambiae and Anopheles arabiensis. So as Nora explained, these are the most important malaria vectors in Africa. These two species occupy very similar ecological niches, but they also have some important difference. Anopheles gambiae mostly feeds indoor and on people. So here I will use the bed nets because this mosquito feeds indoor where there's bed nets. So I use the bed nets icon to symbolize this species. For Anopheles arabiensis, its primary feeds outdoor, and humans and animals, actually the cattle is the primary blood source. So here I use the cattle symbol to represent this species. So in addition, these two species, there's some ecological difference. So Anopheles gambiae prefer the humid environment, and Anopheles arabiensis adapt to the arid environment. So because of these behavioral and ecological differences, the two species may have different landscape factors driving genetic connectivity. So we collect mosquito larvae from 13 study sites across Kenya, including coastal Kenya, Rift Valley, and Western Kenya. So in this study, the Anopheles gambiae, the primary restrict to the Western Africa. As the intervention intensified, Anopheles gambiae become less common, but Anopheles arabiensis is more common. So we have studies population from across the country for Anopheles arabiensis. So due to potential genetic relatedness of larvae from individual habitats, we use the maximum 5 larvae from one habitat to reduce potential bias from using full sibling mosquitoes in the analysis. To measure genetic relatedness, we use the microstarter markers. Microstarter are short repeats in the genome that are generally highly polymorphic. Because they are variable in length, so we can genotype them by electrophoresis, just measure the length of the PCR product. In this study, we genotyped studied over 600 mosquitoes with 5 to 6 markers. We first invested population structure using fixation index, FST, statistics, and the model-based cluster methods known as structure, which assign individuals to ancestral population or clusters. Here, for Anopheles arabiensis, we observed significant genetic structure. So FST was estimated 0.21, and we identified 3 different clusters. So on the left is the population level assignment of ancestral assignment. So the color indicates the proportion of ancestral or cluster membership. So here, on the right here, individual level ancestral assignment, with each horizontal bar indicate individuals. So and the individuals are grouped by population and by the region. So for Anopheles gambiae, we over the relatively smallest spatial scale in Western Kenya, we also found a significant genetic structure. FST was estimated 0.12, and the structural analysis identified 2 ancestral clusters. So we then test whether mosquito population genetic structure is consistent with isolation by distance model. The results shown here are that neither Anopheles arabiensis or Anopheles gambiae conform to the isolation by distance model. So these results suggest that factors aside from geographic distance driving the genetic structure. So then we're interested in what are these factors. So we test the association between population structure and the ecological variables using pairwise genetic distance and the grid layer of ecological variables. We hypothesized to influence gene flow and the GPS coordinates of sample locality. So here we evaluate 6 environmental variables, including temperature, precipitation, percent tree cover, cropland, human density, and the root proximity. So all these data come from public database. So here, this is example of the grid environmental layer for precipitation. So as shown here, in Western Kenya highlands, the rainfall has more rainfall, and then in the Rift Valley and east side of the Rift Valley, the rainfall, the precipitation has the lowest amount. So a link-based statistical analysis with linear mixed effect model was used to determine the effect of these landscape features on the observed genetic structure. So a bootstrap analysis was conducted to calculate model fitness for each ecological layer. Here's the results. For Anopheles arabiensis, we found that the cropland is the top model that explains the population genetic structuring, as evidenced by the average dot arc value, and the frequency of the model was the top ranked model from the bootstrap analysis. For Anaphylid gambiae, tree cover was the top model. Then we can take a close look at the highest performing model to see the relationship between ecological variables and the landscape resistant to gene flow. So in this figure, the y-axis is the resistance to gene flow value. The smaller the resistance to gene flow value, the higher gene flow. For this Anaphylid arabiensis, the area without with crop shows has the gene flow, so promotes gene flow. The areas without crop, no crop, inhibit gene flow. For Anaphylid gambiae, there was an intermediate amount of tree cover associated with gene flow promotion. So too low tree cover or too high tree cover will prevent gene flow of this species. Yeah, we can then leverage this relationship to identify potential migratory corridors between populations based on their ecological relationship. Here areas which likely to have high gene flow as marked by the yellow color. So based on this highest performing model. Here, based on our projection, Anaphylid arabiensis has many migratory corridors in Western Kenya because this population is more widely distributed. So for Anaphylid gambiae, here the migratory corridors are fewer or less due to its limited distribution. So information on the potential migratory corridor could be useful to for developing rational strategies of genetically modified mosquito release. The implication of this study is that human-induced environmental modifications enhance genetic connectivity of mother vectors. So particularly deforestation and agriculture. So because these environmental modifications are actually often associated or accompanied by human settlement expansion and mosquito range expansion. The study limitation, there are a couple limitations with this study. First, we used genetic data from a single time point. The spatial trend may be or may not be consistent across time. Secondly, we only examined the relationship between genetic distance and ecological variables at a one spatial scale. So patterns driving gene flow for this species may vary across spatial scale. Third, the genetic data we used is only based on 5 to 6 microsatellite markers per species, which provide a very limited sampling of the genome. So this for our future study, we want to address this limitation by using more markers with a genome-wide coverage and sampling more mosquitoes and from larger geographic areas. So take-home message. We've detected significant population genetic structure in Kenya for both Anaphylid gambiae and Anaphylid arabiensis. So landscape genetics approach identified ecological factors promoting population genetic connectivity. For Anaphylid gambiae, it is moderate tree cover. For Anaphylid arabiensis, it is croplands. So these factors are actually associated with human settlement expansion. So overall, I hope that our approach that combine population genetic analysis with ecological studies can provide more information on the biology of malaria vectors, and this information is useful to inform the implementation of new vector-borne control measures. So finally, so the study I described here was part of the PhD thesis research of my students, Elizabeth Henning-Schubert, who graduated very recently. So I want to thank my staff and particularly the collaborators from Kenya for their help with the project. So this research is supported by grants from NIH. Thank you so much for your attention.
Thanks a lot, Guiyun Yan, and thank you, Nora, as well. And really interesting talks from both of you. And we do have some questions that are coming in, and I'd like to start with you, Guiyun Yan. Yusuk Lee makes a point or has a question. The FST approach could be problematic in regions where any given location can have multiple genetic clusters with limited gene flow. Do you have any suggestions on how to study such populations for landscape genomics?
That's a great question. Well, yeah, I think for the landscape genetics, we have to look at populations, multi-population as a whole. So not only individual regions. So as I showed in that one, the diagram. So once you have the population genetic structure, genetic variation onto the map, then you overlay with the geographic landscape features. Then actually you can tease out the impact of individual ecological variables towards the population genetic structure. So you can do that one. So even though you have a structure, within each region, but you put the population as whole, you can still tease out the impact of individual factors. So that's come down to we like to sample more populations, so and to tease out to have better resolution on the impact to tease out the impact of landscape features on the genetic structure.
Nora, I'm going to give you one of the questions here. Nikki Faber has a couple of questions, actually. And first of all, she says it was a great talk, and it's blowing my mind a bit. So she's very excited about that. So that's good. So thanks, Nikki, for your enthusiasm. So she says, and you might be reading it too in the chat. Based on the 5 million years estimate, can we make a map of which species have the potential to hybridize? I mean, could you use that as a general? I guess the question is, could you use that as a general tool for predicting hybridization?
Oh boy, that's a really open-ended question, and so. If we're focused on anopheline malaria vectors, as opposed to any species that can hybridize. Let's see. Boy, it's really such a tough question. First of all, this 5 million years is pretty. I wouldn't take that too terribly seriously. It's just so hard to say. We think we're not very well informed when it comes to phylogenetic relationships among anophelines. So you'll recall my having said, the closest known relative to, there could be other species out there that we simply don't know. So with respect to the gambiae complex, and I've consulted Ralph Harbach about this, Anopheles christii is thought to be its nearest relative, and it's more than 5 million years diverged. So in that example, if we're not missing anything, I think the evidence available to us suggests that any productive introgressive hybridization outside of the complex is very rare. But the gambiae complex is one of the best studied, and to sort of ask that question, even of funustis, was difficult. But I think as you move into other complexes, less and less work has been done. So that's just a really difficult question to answer. My gut tells me, just if I can take what we think we know now and extrapolate, my gut tells me that hybridization, introgression, I should say, outside of, from something within a complex to outside is going to be very unlikely. But that's just my gut saying that without data. So I guess the bottom line, I'm rambling and forgive me, but I think the bottom line is that we lack some of the data that we need to do this kind of extrapolating. So can we make a massive map? Probably not. To wrap that up.
Okay. Yeah, good. Thanks. I'm going to give you another one here. This is from Hector Comata, and then there's a comment from Graham White that follows this, which I think is probably going to be it will be worth reading afterward. But anyway, Hector asks, within the gambiae complex, how much introgression goes on between different populations? Okay, and what are the concerns about the release of a mosquito genotype, either GM or non-GM, where it is not native? For introgression between species in the gambiae complex, would there be concerns, apart from a transgene, of exchange of genes between species? So, and he says, I realize this is not a simple genetics question, but so I guess, I think if I'm understanding the question correctly, are there other genetic traits that could be exchanged between these species that would be worth thinking about?
Boy, is that a tricky question. So, it makes a difference, GM versus not GM, first of all, but here is how I think about this, at least as far as the gambiae complex is concerned. The vector species are highly polymorphic. So arabiensis, gambiae, colucii, and very widespread. The other species are much less so and very restricted. So first of all, I'm not saying that introgression can't occur between the vector and non-vector species. It can. The frequency is going to be much lower just because of the numbers game. And I think this gets back to a question that you haven't asked me yet, but you're probably going to. I think the direction of introgression may matter. So I think the reason that gambiae, arabiensis, and colucii are so widespread across the landscape is because of the variation that they carry in their genomes that confers the ability to be competitive as larvae in the larval breeding sites. The other species are limited probably because of a lot of competition from the more dominant vector species. So I would be less worried about gene flow from a non-vector into one of the primary vectors than perhaps the other way around. So I'm not sure if my answer is making sense. I can't tell. I can't see an audience. It drives me nuts. This whole Zoom thing.
It was a tough question.
The issue is, okay, a hybrid forms, right? You've got an F1 hybrid, and the female, regardless of the direction of the cross and which species are intermating, the female is going to be presumably not only viable, but vigorous and fertile. But the issue is, is that individual going to be competitive in the environment? And I think that depends on the direction of the well, first, is it going to survive? Then the direction of the back cross. And yeah. So sorry, I'm rambling again.
No problem. So no, there was a comment that was made. Yeah, there's a comment that made, Greg Lanzaro just made a comment that that related to Hector's question, and it just brought up the evidence that he had reported on in the past of the insecticide resistance gene KDR introgressed from gambiae to collucii. And so I think that was, maybe that's an example for that addresses some of what Hector was asking about in terms of other genetic characteristics that have that introgress that are of some significance.
Oh, sure.
Yeah. Okay. Super. I wanted to get to John Mumford asked a question or makes a comment in a question, which and John, as you know, is on Target Malaria, involved with the Target Malaria initiative in some ways. Much of the concern, he says, much of the concern about introgression is, I think, about a gene drive for gambiae having a negative impact on non-vector species in the complex through entry into those populations. Do you see any concern in reverse about introgression from the non-vectors in the complex, introducing any potential resistance to the gene drive, for example, mechanism, the resistance mechanism, into the central target group in the complex? For example, some tendency for assortative mating or other behavioral isolation. So there's another tough one for you.
Yeah, it's really tough. There's a lot going on in that question. So with respect to resistance, I'm interpreting that, and I may be misinterpreting that, to mean resistance to the drive by virtue of an allele coming in that the gene drive doesn't recognize. I mean, that could certainly happen, and that would be a risk. As far as assortative mating, that's a whole other issue. Which is a pretty tough question. To my mind, it's hard for me to imagine. Sorry?
I think those are just sort of, he was just giving some phenotypic examples of things that might occur. That the things that might come from a non-vector species into a vector species that, okay.
I mean, it's hard for me to even understand why these hybrids are back crossing, because presumably they should be a little bit messed up as far as their mating preferences are concerned. But it's a little difficult for me to imagine that a complex trait is right. If it's not a single gene derived trait, if it's a complex trait where multiple factors across the genome have to come together to sort of make that trait happen, it's hard for me to imagine how a rare hybridization event is going to sort of transfer a different trait into Anopheles gambiae from another species, unless there's a tremendous amount, not just a very rare hybridization event, but sort of like intense hybridization for I have no idea what reason. So I personally think I may have missed something, and others in the audience can jump in, but I think the chances of that are rare. The Greg Lanzaro's point about when we talk about KDR, that's a single gene. And yes, there's a huge amount of selective pressure for something like that to transfer among species that are going to be exposed to the insecticide. So there's a very clear example of a single allele transferring easily. But when you're talking about a behavior that has a really complex genetic basis, the chance of that coming over in a rare hybridization event intact, so that the trait transfers is, I think, minuscule.
Mm-hmm. Okay. Hey, thanks a lot. Guiyun, here's a question from Nikki Faber, who is also very enthusiastic about your talk as well, and some of your your slides, which she considered to be artwork. So thanks, Nikki, for her enthusiasm. And her question has to do with, she has a couple questions. So I'm going to go to her second one first. What is a relevant scale? What is the relevant scale of migratory barriers in mosquitoes? Only the huge ranges like the Rift Valley you mentioned, or are there smaller ones also?
Yeah, from our study in Western Kenya. So if I can go back to, let's see, this one. So for arabiensis, so it's like West Kenya is almost like a one population, right? So, but then we see West Kenya, Rift Valley, there's sort of some isolation here. But for Anopheles gambiae, this is based on our model, based on our analysis, shows the population are more isolated. So if from here to here is about like 40, 50 kilometers. So this shows sort of like isolation here. So over here, I would see geographic scale. It's really depend on the population, depend on the locality, depend on local ecology. So it's really difficult to generalize that.
Mm-hmm. So that map there is very suggestive of that one village along the lake on the right side of that would be somewhere that looks fairly isolated from the other populations.
Yes.
And I guess if you were planning a release program or something that involved a release of mosquitoes, that would be something that you'd probably want to know, I guess.
Yes, that's right. But I think it will be nice that we do additional sampling in the area, so do high density sampling more populations, so we can test that out better to figure out the impact of these landscape features on the gene flow. So right now we have limited number of populations here. Would be nice to do high resolution.
Yeah, great.
Yeah.
Mm-hmm. Nora, could you say something about, you mentioned in your talk, if I heard it correctly, you mentioned a region where there was a lot of hybridization that was exceptionally high, that was sort of, you sort of took it as an outlier and put it outside of an analysis that you were talking about. Could you say something else about that? That sounds fairly intriguing.
It is a part of I refer to it as far West Africa, where for reasons that are, as far as I know, not well known. But the rates of hybridization between, if Greg Lanzaro is here, you should make him answer this. But anyway, where the rates of hybridization are way up there in the multiple percents. And you know, I'm not sure that gambiae and Colutzii have collapsed as independent entities, but depending upon how you want to define them as separate gene pools, it's pretty close. Really, that this touches closely on some of Greg's great work. He's looked closely with markers at this, and you know, I honestly can't remember if he found total collapse even in what are normally very distinct regions on the X chromosome or not. I mean, that's all I can really say. As far as I know, no one has nailed down what the ecological factors are that are underlying that type of breakdown.
Mm-hmm. Okay, we'll have to get him to give a webinar on the Gene Convene webinar series, so he can talk about his work. Greg, I know you're out there and you can hear me. I have another question, Nora, that's really a technical one, I guess, is could you say something a little bit more about this Abba-Baba methodology, just not pretty much, if you could conceptualize about the basis by which, you know, you're making these determinations of introgression without getting maybe too technical, because I'm not going to be able to understand it, but just maybe conceptually how that methodology works.
Okay. So, oh boy, let me do my best here. So when you have very recently diverged species, they start from a common gene pool, right? And then they separate. And we like to think that there will be fixed differences eventually when you compare different lineages, if enough time has gone by. But very soon after the separation, there will be shared, what we call shared ancestral polymorphism. These are polymorphisms that segregate in the descendant lineages. So if there's no introgression, so the shared polymorphism simply goes down one avenue or the other randomly, there should be no pattern discernible about how those alleles segregate. It really should be stochastic. If there's been an introgression event, that should systematically change the pattern. So again, without going into detail, there's a very simple formula, but I don't have a blackboard or a whiteboard. So the bottom line is that this test picks up a skew from what would ordinarily be just a stochastic pattern in the way that the ancestral polymorphism is arrayed. And this is inferred based on whole genome alignments, where at each alignment position you have, a variant. So I hope that sufficiently answers the question.
It begins to give me, it does begin.
About how good of a test this is about hybridization. It's important to stress that, it's looking at historical introgression, not contemporary. And like any test, it's not perfect. So, it's just an indication, and it has its assumptions which can be violated. If you carry out a test like this on species that are too distantly related, you can get false results. So all it is an indication.
Great. Well, we're coming to the end of our time here, and I'm going to start to wrap things up here. And I want to just, first of all, thank both you and Guiyun Yan very, very much for participating in our in the seminar series that we have. It's really great and very informative and thought-provoking. So I just want to say thanks a lot for doing this.